Good morning and welcome back to our short talks session for this morning. We'll be the same format as we've had all the rest of the week. Speakers have 8 minutes to give us a little short talk, followed by 2 minutes of questions. You can find the microphones on the sides. Please do use them if you shout out from the audience. It may not be as audible to the online viewers what your question is. For speakers, again, reminder, I'll be sitting in the front and I'll kind of raise my hand when you have about 1 minute left. So you know you can kind of wrap up if you accidentally got on a tangent. I know I do. And with that, we'll jump right in with our first speaker, Pascal Belleau, going a computational framework for inference of genetic ancestry from challenging human molecular data.
Yes, thank you. So I'm Pascal Belleau for the Graduate Lab at Cold Spring Harbor. We will talk about the packaged RAIDs that infer the genetic ancestry from genetic ancestry for challenging molecular data. It's well known that some diseases are associated with ancestry. Example for cancer, prostate, you have a higher incidence in the African American population. In leukemia, you have a higher incidence in the white population. And for the cancer, the thyroid cancer, you have a lower incidence in African American population. The association is probably a mix between environment factor, genetic component, and a lot of stuff, but we are particularly interested in the genetic component, genomic component, and in this case, it's important to have the genetic ancestry call, not just the self-report ancestry. So it's well known how to infer genetic ancestry from the standard genotype from cancer-free whole genome, whole exome, and you have methods like clustering following dimension reduction, PCA, to do it, or likelihood maximization for the allele population frequency, like implemented in the admixture. But in our case, we have a double challenge. We work with cancer-derived molecular data, so without the normal genome, the cancer-free genome, and with other protocol like RNA-seq, ATAC-seq, without the whole genome or stuff like this. So if we have access to call the ancestry on this type of data, we get a lot of public database to do the analysis. So we develop a method to do this kind of inference. We publish it in the Cancer Research in 2023. And currently, the RAIDs package is in bioconductor. It's called the global ancestry, continental ancestry, for the whole genome exome sequence. The gene panel, like one, I forgot the name, RNA-seq, single-cell RNA-seq, ATAC-seq. We are working now for the next release, include the admixture, and probably later the local ancestry, low coverage, whole genome, citizen converted sequence like for methylation. We use a lot of package from bioconductor, genomic range, GDSFMT, SNP related. So the package use first a reference. We need a reference population. We use by default is 1000 genome, the new version, but we can use the reference you have if you prefer. We use the 1000 genome because it's public and not restricted. So the idea behind RAIDs, we start with the reference population. We extract variant with some cutoff frequency for at least one of the population. We extract a subset of this reference sample, a subset of samples from the reference. We generate what we call the synthetic data. I will explain a little bit more about the synthetic data. and we optimize the parameter for the method, depending the methods. The method is a module, so it can be PCA with a key neighbor, or UMAP with something else, or a mixture. So if we use PCA, for example, we have to select the number of dimension and the number of neighbor to do the inference. So we optimize with the statistic data, and we can use the statistic data to be able to to validate the inference. So if our method is able to accurately infer the ancestry for this type of data. So what is the data synthesis? Sorry. The data synthesis, we use the reference sample with known ancestry. We extract the genotype we have selected at the position we have selected. After we take the cancer profile, for example, we extract the characteristic of the cancer sample, the sequence depth, the allelic frequency if we have copy number variation. So in some case, we have a lower allele fraction for the SNP at this position. And we generate a sample with the genotype, but with the genotype of the reference sample, but with the characteristic of the cancer sample. And we do the inference, the ancestral reference on this sample. So we know if the inference is correct or not. And so we can use it for the measure the accuracy or optimize the parameter. So we use the method on more than 1,000 samples for whole exome sample, panel, and RNA-seq. Here we have the comparison. The accuracy is the comparison between the cancer sample and the normal cancer-free sample. So we compare the ancestry inference, and we have a concordance around 98%, 99% for all except the panel for the pancreatic cancer. It's organoid pancreatic cancer with high cellularity and a lot of copy number variation. This case is a little bit more complex, so we have a lower accuracy. If we look at the performance with the statistic data, for the whole exome, the foundation one is the foundation one, the panel. The RNA-seq, the single-cell RNA-seq, and ATAC-seq, we have around 99%, except a little bit lower for the foundation, because the foundation is just something like 300 genes. And the rest is around 99, 98%. So in conclusion, we developed an accurate method to infer ancestry from challenging method for challenging molecular data. And the data synthesis give us the ability to know if the method is accurate or not for this type of sample. And we are implementing a version with the admixture and working on local ancestry and pangenome. We are extending to the method to the methylation method. Just an overview for the performance admixture. It's go really well on TCGA and really well too on synthetic data on colorectal, except the American population, because American population have American and mixed population because the reference is not well defined. So I would like to thank all the team, Alexander Glatznis, the main PI, Astrid for everything, Astrid Deschaines from Stevenson Lab, Vince Curry for discussion, and the funding organism, the clinical collaborator from a lot of people. Thank you.
I actually have a question about how you handle RNA editing when you're looking at RNA-seq data and trying to call ancestry from that data.
Yes, we use exactly the same method. We call the SNP, depending of the method, We have multiple ways to call the SNP. And at the position, we check for allelic, specific allelic expression, if we have a difference, and validate with synthetic data if we are able to call the ancestry on this data set. And if we are able, if we have a good way to do it, we call it.
Okay. And if you can't, it's just not decisive, right? If you can't identify what the original genotype was.
We don't need to know what is the genotype. No. Okay. Is the reason we use the synthetic data. So we use the reference, a subset of the reference. We remove a subset of the reference. We use this genotype with the characteristic of the RNA-seq data set, and we use it for validation. And if the data set, and normally for RNA-seq, it works pretty well. Normally, maybe some exception if you have a pathological sample, but normally it works very well for the RNA-seq. And we don't need the exome or DNA for do it.
Great. And a quick second question is on the batch effects. You're working with various data sets, and especially PCA is prone to those batch effects for ancestry determination. What do you do with that? How do you deal with it?
What do you mean by for the batch effect on the ancestry card?
Yes.
Yes, depending. If probably we have to adjust if we compare RNA-seq with exome or something like this.
Even between RNA-seqs, like if you're doing from one side versus another.
Yes, it's not so bad if the RNA-seq, if they are relatively, the effect on the SNP call is not so high. It's not like the expression. The batch effect is a lot lower than the expression level.
So comparable to...
Yes, it's easier to compare. Of course, the level of detection of the SNP is not the same if you use a whole exome or a whole genome and RNA-seq. But for RNA-seq, between RNA-seq with relatively similar stuff, the batch effect is not too high.
Okay, thanks.
Thanks. Okay. Thank you.
Thank you so much for that talk, Pascal. We're moving on to our next speaker. Whoever's moderating online, can you just raise your hand? I didn't read. All right, cool. You'll shout out if there's online questions? Sweet. All right. Brings us to our next short talk that is recorded on Exvar, an R package for gene expression and genetic variation data analysis and visualization. This is presented by Hiba Ben Aribi, and we will get that started for you in just a moment.
Hello, I am Hiba Ben Aribi, a graduate student from University of Tunis in Manar, Tunisia. I will present today an R package, named Exvar, developed in collaboration with Imran Dixon, Najla Abbasi, and Olaitan Awe. RNA sequencing is a fundamental technique in biological research, providing comprehensive insight into gene expression and regulation. However, despite its importance, RNA sequencing data analysis presents several challenges, especially for beginners and non-data scientists, due to the overwhelming multitude of tools, pipelines, and programming language available, which requires a significant amount of experience to navigate effectively. To address these challenges, our team decided to develop a comprehensive RNA sequencing data analysis pipeline. We chose R as our programming language due to its extensive ecosystem of bioinformatics packages. The workflow covers all major steps, making it accessible and user-friendly for researchers at all levels of expertise. Instead of reinventing the wheel, we focused on utilizing existing open-source packages that have been thoroughly reviewed and validated by the scientific community through peer-reviewed publications and those submitted to the CRAN and Bioconductor projects. In total, we integrated various functions from about 70 packages into a single pipeline. And we wrote custom code to fill any gaps and ensure the compatibility between the inputs and outputs of the different packages. We managed to cover the entire workflow from FASTQ files preprocessing to data analysis and finally data visualization. As our pipeline grew to 1,000 lines of code, we recognized the need to make it more user-friendly. We divided the pipeline into manageable functions, transforming it into a novel R package. We name it Exvar. It has 10 functions. 1 for installing all dependencies, 1 for FASTQ files preprocessing, 2 for genetic expression data analysis, 3 for variant calling, including single nucleotide polymorphism, insertion and deletion, and copy number variation, and 3 functions for data visualization. The visualization functions are implemented as Shiny application with a graphical user interface. We also extended our pipeline to support multiple species, including the human species and 7 model organisms, which are the mouse, the rat, zebrafish, mustard plant, yeast, roundworm, and fruit fly. The package is currently available on GitHub. We made sure to document it as much as we can by including tutorials, package user guide, demonstration data, and a case study. The first step to use Exvar is to install it using the DevTools package. Next, you need to install the dependencies using the requirement function. The list of supported species will be provided in the console for you to define your target species. The next step is to preprocess the FASTQ files using the process FASTQ function. All the functions are well documented, and you can use the help feature to understand the parameters of each function. Once the BAM files are generated, you can start the gene expression analysis, using the count function to get the gene count data, or the expression function to define the genetic expression profile. The 2 functions will generate CSV files. Later, you can use the visx function to visualize the data. Once you run the command, the Shiny application graphical user interface will automatically open. You can investigate the gene count data quality, define differentially expressed genes, and visualize them as volcano, MA, or PCA plots. Additionally, you can perform the gene ontology enrichment analysis and visualize the result as dot, bar, or CNAT plots. You can use the call SMP and call indel functions for single nucleotide polymorphism and insertion deletion variant calling. The 2 functions will generate VCF files. You can use the visSMP function to visualize the SMP data and compare the polymorphism between the study groups. Finally, you can use the call CMV function for copy number variant calling, which will generate VCF file as well. And you can use the visCMV function for the CMV data visualization. By identifying the recurrent CMV region, visualize the overlap between the CMVs and the genes as oncoprint plots, and performing the overlap permutation test to assess statistical significance. As mentioned previously, the package is currently available on GitHub and will soon be submitted to Bioconductor. If you wish to contribute to the package development, feel free to do so via pull request on the project repository. If you have any questions, do not hesitate to contact me and the code developers via email or LinkedIn. Lastly, I would like to thank the Bioconductor Conference organization team for this opportunity to share our work today. Thank you for your attention and time.
So she is available online to answer questions. Are there any questions in the room? Any questions online? All right, I have a question then. So I like this as an all-in-one kind of package instead of, especially for beginners and learners, and also especially when you have like a grant due on like Monday and it's 10 p.m. on Sunday. You just need a quick, analysis to throw in there. How easy is it to plug and play other RNA-seq, modules and throw in your favorite gene set enrichment thing and plug and play in this thing within your pipeline? You'll have to unmute if you are in the little space. There you are.
Hello, everyone. Do you mind repeating your question?
Yeah, just how easy is it to plug and play other RNA-seq packages and modules in the downstream and in the mid and with your pipeline? Can you just like throw in extra little bits?
Yeah, so the package is available open source. We tried to make it like it is complete for the workflow. So actually, if you don't want to integrate any more packages into the pipeline, it is sufficient. However, the output and the input of all the functions are the standard and the most common used data files. So it is easy to integrate any novel feature into it. And we have a full PDF detailed with the detailed explanation of how to integrate it into your pipelines, and it is available on GitHub.
Thanks. Another round of applause.
Thank you.
Our next speaker is also pre-recorded and online, unraveling the intricate molecular landscape and potential biomarkers in lung adenocarcinoma through integrative epigenomic and transcriptome analysis. If you guys are in the app, they have a really good visual abstract in the app as well to go along with the talk. So if you're, if you want to have that image, it's there. And this talk is by Arnab Mukherjee. I hope I probably butchered that too, but thank you.
Hello everyone, welcome to my talk. I am Arnab Mukherjee, PhD student at Manipal Institute of Technology, India. Today I will be speaking on unraveling the intricate molecular landscape and potential biomarkers in lung adenocarcinoma through integrative epigenomic and transcriptomic data analysis. According to the data for the year 2023, lung cancer remains a prevalent type of cancer and is expected to be a significant contributor to cancer-related fatalities globally. Non-small cell lung cancer, specifically lung adenocarcinoma, accounts approximately 80 to 85% of the reported cases of lung cancer. Multi-omics provides a holistic view of the interaction between genomic, transcriptomic, proteomic, and epigenomic factors. This integrated perspective aids in unraveling complex molecular mechanisms driving cancer development and progression. Tobacco smoking has the most significant influence on the lungs, being accountable for 87% of lung cancer deaths associated with the alterations in the DNA methylation at the CPG sites. Lung carcinogenesis unfolds in a sequential multi-stage pattern, gradually developing genetic and epigenetic alterations. The epigenetic alterations induce the development of malignant characteristics and aggressive traits in lung cancer cell and acquire resistance to conventional therapeutics by dysregulating multiple signaling and metabolic pathways. Enhancers govern gene expression across great distances by looping DNA and offering distance regulatory region closer to the target, the gene promoters. Therefore, we employed this ELMO analysis to determine the enhancers and link enhancer status with the expression of the target genes to discover transcriptional targets. So where we use DNA methylation and transcriptomic data sets, which are incorporated for the ELMO analysis to identify the probe gene pairs, transcription factors, and motif enrichment. The data sets of the transcriptomics and the methylation, DNA methylation were acquired from the TCGA database and gene expression omnibus using a bioconductor package called TCGA biolinks and GeoQuery. And the differential expression was carried out using DSIG2 tool and Exvar tool. The differential methylation was achieved using the CSAM package. These datasets were subjected to the ELMA analysis, which was achieved by creating a multi-assay experiment object and separated into primary tumor and solid tissue normal samples. The overlapping differentially expressed genes was identified from the TCGA data sets and the gene expression omnibus data sets, subjected to the network construction and analysis of the topology, identifying the critical hub genes. followed by identifying their biological processes in lung cancer progression and the clinical significance in lung adenocarcinoma using a survival analysis. So the differentially methylated sites, CPG sites, were identified, uncovered using the Wilcoxon test, and the differentially expressed genes of the adjacent probes were identified. Further, the distal probes signifying the regions enriched with these enhancers were identified by based on the HG38 human reference genome file. For each distal probe, the methylation levels were systematically sorted across all the samples within the primary tumor and solid tissue normal samples. Considering the smoking status of each group, this analysis tested inverse correlation between each probe's methylation level and the corresponding gene expression, which was achieved using a Mann-Whitney test illustrated in the form of a heat map. The differentially expressed genes between the TCGA and gene expression omnibus, we identified the 419 genes which were in common. Further, we studied the physical and functional interactions between these differentially expressed genes using a protein-protein interaction network, which from which a notable cluster was identified with the highest score of 16.667, and a maximum click centrality algorithm aided us in identifying the top ranked top 10 hub genes within the cluster. Further, we identified notable changes in the paradoxical overexpression of the ribosomal proteins, which were found to be, their CPG sites were found to be hypomethylated, and the mitochondrial ribosomal proteins, which their CPG sites were hypomethylated and were upregulated. The transcription factors were responsible for regulating enhancer-associated hub genes, which were predominantly linked with the hypomethylated regions. So we determined the site-specific transcription factor binding motifs and were enriched within the enhancer-linked hub genes in the form of a directed graph. And this transcription factor binding motifs were investigated at the 250 base pair up and downstream of the human 450K platform, for which were illustrated using the odd ratio greater than 1.1 as significant ones. And the top 5 were listed for the hub genes which we identified. The gene ontology analysis identified the gene ontology terms are describing their biological processes. The total fold change enrichment analysis for the hub genes indicated the ribosomal proteins that were paradoxically overexpressed and hypermethylated, enriched in the process related to impaired rRNA processing and ribosome biogenesis, which was active in a cell cycle dependent manner, promoting cell proliferation. The overall survival of the expression cohorts in lung adenocarcinoma patients was analyzed for the hub genes over 200 months, that demonstrated the high expression cohort of the genes were associated for the lung adenocarcinoma progression, which was indicated by a hazard ratio greater than 1. So the hypomethylation disrupted the mitochondrial translation processes, which affected the assembly of oxidative phosphorylation complex, and consequently the mitochondrial function. The hypomethylation which led to the altered cellular metabolism and might increase the reactive oxygen species in the cancer cells and a compromised electron transport chain. Further, the elevated ROS might contribute to the genomic instability and resulting in suppression of tumor suppressors and inhibition of cell cycle arrest and promoting cell growth. This is the model which we generated based on our findings, and we thank ComBio 2022 for providing the Jetstream 2 resources to carry out this work, and Manipal Academy of Higher Education, Manipal. These are my references of the study. Thank you for attending my talk, and you can contact me anytime using in this email and you can contact. Thank you.
Unfortunately, he's not here with us online, so we can't take any questions, but he might be in the Slack later. If you do have anything, you can ping him there in the group Slack. Our next speaker, whoops. Oh, he was on time anyway. The next speaker we have is Myriam Maumy, patterns deciphering biological networks with pattern heterogeneity, you know what I'm trying to say, multi-omics measurements.
Hello. So obviously I'm not Myriam, but because of some contingency, I will give the talk instead. This work was done with biologist and medical doctor, and it was in order to help to decipher some mechanism behind leukemia. So basically, we had to deal with two kinds of measurements from a statistical point of view and from a biological point of view. So we had RNA-seq data and also proteomics data. So after using several kinds of filtering, either independent filtering or gene expression-based filtering or according to the abundance of the protein, we selected a few thousand genes, expressions, and proteins that were from a dataset that had a time course measurement that was designed to compare the evolution of leukemia between two groups of subjects. So basically, we had the baseline time t equal to 0, and then we just tried to decipher the way the signal will propage through time. So we deal with special experiments that are targeted experiments that can be viewed as cascade of transcriptomics and proteins. And basically, we wanted to design a special inference technique in order to take into account the fact that you have this cascade. So basically, you have some actors that can be either gene or proteins that can act on to each other at any time further. So basically, at time t equals 0, you have the reference. Then you stimulate the cells in the two groups, and you compare the way the cell will evolve through time by using those two kinds of measurements. So we designed several kinds of interaction in this particular case, but the package can be used for more general settings, of course. And so basically what we had to find was a problem that is twofold. You have first to find the structure of the network, which is a gray network on the graph, and then to find the way that the signal will propagate itself in the network. So this is the way that the network will lit up after the stimulation and at time 0. Okay? And this way, we were able to find some subnet that was especially associated with the leukemia that was studied at the hospital. Okay, so basically what is the problem, one of the main problems in that kind of data is that you got noisy, highly correlated, and high dimensionality. But since you use a targeted cellular process with that simulation that synchronizes the cells, you can use take this as an advantage in the model. So we wanted to use also some a priori knowledge and some sparsity, because you know that it is very important in those kind of models. And so we wanted to be able to perform weighted inference to take into account some different level of certainties in the a priori knowledge, because we dealt with cancer cells, and so what is known for regular cells is not always true for cancer cells. So basically, we had a multi-level. We cluster the gene expression into clusters, and the clusters will be able to act on each other through time. And we also add some information at the individual level to know whether what actor can act on other actors. So the clustered expressions were those ones. So you can see that we had several different patterns through time. And we decided that given a cluster, it can act on other cluster after the moment that it has moved from 0. So this way, you can design some specific matrix in the model to enforce the temporality. And you can, this way, highly reduce the number of parameters in the model, which is very helpful because we had few measurements. So basically, through time, inside a cluster, you can have this way of acting between inside the cluster through time, and between the other cluster, you have the same thing also. Basically, at the individual level, we just integrated some transfer factor data and sparsity and so on. Basically, the model equation will be this one at the end. So it was one kind of linear model, and you integrate both the omega, which tells you if any actor can act on any other one at any time, and the F1, which can model the way the signal will progress through time. So the result of the inference will be twofold. And this way you get quite a versatile modeling tool because you can separate the inference of the structure of the network and the inference of the signal through the network. So basically we use some dedicated estimating method, and well, we were able to estimate the F matrix, the omega, and when you combine the F matrix and the omega, you get the evolution through time, which was depicted on the one of the graph at the beginning. You know, the graph with the gray lattice. So the gray lattice is found using the omega. You threshold them, and the ones that are slightly non-zero will give you the gray lattice, and then integrating the F matrix that were inferred, you can have the way that the signal will propagate. So this is what was quite hard in this reverse engineering problem, is that you have to infer both the structure of the network and the signal itself. Okay. So that was why we created this technique. And we did some benchmarking, of course, and the package and the technique itself improves over other existing methods, especially because we use the fact that we have time-related data and this cascade technique. So if you have that kind of periodic data or cascade network or even pattern data, feel free to use the package. It's available on the Quran, and it should improve your results. And if you want to know more about this, you can just go to the website dedicated to the package. You will find several vignettes and examples online so that you can use it yourself and try to get into it at a slower pace.
Hi, this looks really interesting. If I have an estimate of let's say, expression over time as a continuous function, is that something that I could input into a framework like this to do a similar inference of?
What do you mean by continuous over time? You have what's the frequency of the sampling? It's a high frequency?
It's samples that are irregularly sampled from different people, and so they're not lined up into well-defined time points.
Yeah.
And so instead, we basically get a smooth estimate of the changes of expression over time.
Yeah, that's what we did in that experiment. We had 9 time points, but it was we had a longitudinal data set with 9 times, but I don't know how many times you got in your one. But the more time you get, the more you will have the cell F matrix increase their size. So basically, it will depend on how many time points you get per subject.
I guess the other way to put it is if I have time points from different subjects that are not lined up.
And not lined
let's
up.
say it's in a clinical setting.
So if they are not lined up, it will be more tricky. More tricky. I think that it is designed to work with regular time. Yeah, but maybe you can infer the function and then sample.
That was a really good question. We should chat at lunch. I have my clinical trial data is the same because patients they sometimes miss appointments. They get sick. You don't have a time point for every person at the same time point. And I'll share some math we have for that. Our last speaker of the day, of the session, not quite ready to go home yet, is Kelly Street from University of Southern California. His talk will be Multi-omic analysis reveals phenotypic plasticity in colorectal cancer.
Thank you, everybody. Thank you for having me. I'm going to talk about a really cool experiment that we did in colorectal cancer. And I just want to start off by giving a huge shout out to Daryl Shibata and Kim Sigmund, who brought me into this project when I started at USC 2 years ago. Daryl is an MD who works on colorectal cancer. Kim is another professor of biostatistics whose expertise is in methylation data. And so I sort of got a little bit of hands-on experience with methylation data, which was not something I had played around with a whole lot before. And then Yifan is a PhD student in epidemiology who's been working with me on this analysis, and she's fantastic. So plasticity is sort of the unifying theme here. And according to this paper that I found, it is the ability of intestinal cells to adopt an alternate cell fate in response to a perturbation, including dedifferentiation. This is a really interesting sort of characteristic that some cells can have that basically allows them to change their type, change their expression, without changing the methylation conformation or other sort of epigenomic features that we typically think of as controlling gene expression. And so this is what that looks like in a colon crypt. So this little U-shaped structure is a crypt, and you've got a bunch of them in your colon. And basically down at the bottom of the U, there's a population of stem cells that can differentiate and sort of move up the two sides of the crypt and form all the different necessary cells in your colon. I'm also going to give a quick shout out because I showed differentiating trajectories to slingshot, trade-seq, and condiments, which are like my previous life of doing all sorts of trajectory analysis stuff. Didn't do that for this project, but I did bring stickers. So if you haven't visited the front desk since day one of the conference, feel free to go get some stickers. What we did do is we looked at methylation. And so we used 850K epic arrays to measure methylation at several, hundreds of thousands of CpG sites along the genome. And we summarized that at a gene level because we were interested in looking at methylation variability. And so I'm using the term variability loosely here because we only ever had 2 samples. So it's really just a difference. And if you're familiar with the 850K chips, there are a handful, maybe for most genes, 4 or more is where we set the cutoff. But any gene with at least 4 CpG sites in it, we took the beta values for those CpG sites from sample 1 and sample 2. In the case of normal healthy colon tissue, these were from 2 different crypts. And in the case of colorectal cancer, these were from the same tumor that had been cut in half. And so we did a sample from the left side and a sample from the right side. And we basically just compared for every gene the beta values at each of the CpG sites that were annotated to that gene and took the average of those differences as a measure of the variability of the methylation for that gene. This is just a histogram of what that looked like across the 2 different sample types that I mentioned. You see that there's a slightly lower mean in the colorectal cancer samples, and that's because the tumor has more recently differentiated from, a single progenitor than the colon crypts, which are sort of reproducing themselves in sort of a regular pattern. Where I joined this project was identifying a couple papers that had done single-cell RNA-seq in similar systems. And so we brought in single-cell RNA-seq data from normal colon and colorectal cancer in completely different patients, I should note. And I was tasked with measuring the variability of the gene expression at the single-cell level so that we could compare it to the methylation variability that I just described. And this is where things got really interesting. So if you're familiar with Will Town's excellent paper on GLM-PCA, we used the deviance measure from that paper to measure the variability of expression for every gene, controlling for things like patient level and subsetting to specific cell types, like the cell types that are present in normal colon or in colorectal cancer. And basically what we found is this really strong negative correlation between methylation variability and expression variability. So this is definitely not what you would expect under a remodeling paradigm. The genes that have the lowest amount of methylation variability are the ones that had the highest amount of expression variability. So we saw that genes were changing their level of expression without any corresponding change in the level of methylation. We think that this is really indicative of a plasticity paradigm where these genes are able to adapt to the local microenvironment without changing anything about their methylation profile. Again, this is very different from the classical remodeling paradigm that some people, think of when you think of methylation. You think increasing methylation on the promoter tends to decrease the expression of a gene. And so it's sort of methylation comes upstream of expression and it controls how much a gene can be expressed. And that's not what we're seeing here. What we're seeing is basically the tumor and normal colon as well have certain genes that are important for the function and adaptability of a cell to survive in different microenvironments. And those genes are basically left alone in terms of methylation so that they can do what they need to do in terms of expression. And I think this is a really cool idea, and we've got a preprint on those previous results, but I'm really interested in looking at this idea in terms of the 10x multi-omic pipeline. So do we see these same patterns in ATAC-seq and RNA-seq? And it seems like, we easily could. I haven't actually gotten to this point yet, but if we're thinking of sort of an epigenetically permissive phenotype that allows for different types of gene expression, different levels of expression, that definitely sounds like something you could measure with ATAC-seq plus RNA-seq. One of the challenges here, though, is that in order to do this type of analysis, we need to link the features from the ATAC-seq to the features, which are genes, from the RNA-seq by some sort of metric that basically tells us, okay, these are the peaks that might be related to the expression of this gene. And a lot of people will do that by just linking the closest peak to each peak to its closest gene, sorry. But what we want to try is to basically leverage a huge database called ANOQ, which has compiled a whole bunch of results from previous GWAS studies that are for linking SNPs in your DNA, and essentially leveraging those annotations to link peaks to genes based on functional annotations from previous GWAS studies, because there's a ton of research that has already been done on this. And so there's a lot of outside information that we can bring in and sort of leverage in making these mappings between the different feature spaces. And so I would love to say that I have some awesome paper and some cool R package and that I've already done all this, but I haven't. So if you want to collaborate, come talk to me. I've got a little bit of funding for a PhD student, and we've got a T32 if you want to put in an application for a postdoc position. I would love to work together. So thank you very much.
You got there before Tim, so it's all you.
This is really interesting data, and I was just hoping that you could clarify a little bit more where the methylation sites are that you're actually measuring when you get that negative correlation. And so are they in the gene body, or are they specifically at the promoter site?
They're in the gene body. We did it both ways, but the signal was a little bit more clear with the gene body, but we also did it with just the ones from the promoter.
And you see the same effect of the
Yeah.
Okay, that's interesting. Yeah, because the gene, like, what's the mechanism by which the gene body is regulating expression? It makes sense at the promoter site how that regulates the recruitment of transcription factors, but at the gene body, how is that regulating transcription?
I don't know if it necessarily is, but we're looking for essentially how much is the methylation profile changing from sample to sample. And so there are just more CPG sites in the gene body than there are in the promoter region. And I think we got a slightly cleaner signal with that.
Thanks.
Yeah.
Couple of questions. Do you weight by the number of beads detected for a given CPG in a given sample?
We did not.
And then in terms of why there might be variability, is it known whether the oxygen tension in a pre-adenoma is greater or lower than the neighboring crypts? So you have demethylase enzymes that are iron 2+ dioxygenases, and their throughput essentially depends upon the local oxygen tension. So is it possible that this is a metabolic side effect of the evolution of the early evolution of a tumor in a shared local environment? I know that Daryl has some matched bilateral separate origin crypts. Have you gone back and looked against those?
We haven't, but I'd be interested in exploring that. I'm not a medical doctor, so I don't really know what the other possible explanations might be. But yeah, I'd be more than happy to chat about it if you have suggestions that we could look into.
It's a cool project, but it's always been difficult, which presumably is why.
Yeah.
Just keep right on going.
Yeah.
It's a neat talk.
Thanks.
Okay. Thank you. So we now have a 20-minute break. You can stretch your legs, grab some coffee, and we'll meet back here for our next keynote talk. We'll see you in 20 minutes.